Dr. Kinchington, welcome, and thanks for joining us.
Thank you. Thank you. Glad to be here.
Yeah, it's always really good to see you, and I really love your work, and I think it's something that a lot of people may not get a chance to hear enough about or understand. And it is a problem that certainly a lot of people deal with. So you came here. We maybe should get a little bit into your background just a slight bit because you came here from Great Britain and then via the United States to come and start your academic career. But and you decided to get into science and into this world of medicine and science and trying to understand these viruses and how they affect the eye. What was it that led you to make this a career path for you? What are the things that really got you inspired to make this your something that you were trying to do and contribute to science?
So the whole started when I was doing my undergraduate degree, actually, way back in the 80s.
Hmm.
One of the things that I got really excited to do was a project on viruses as an undergrad. And I continued that with my PhD, and that was working with herpes simplex, the virus that causes cold sores and eye problems. And I really fell in love with viruses. They do amazing things. They adapt the host to their growth, and they're always important. We've always had them around, and once in a while they emerge, as we all experienced with COVID, which has just come up. They're emerging, and some of us have been with us for millions of years, such as Varicella Zoster virus and herpes viruses, adenoviruses that cause pink eye. And I just love working with viruses because they really adapt the host cell. They change the host cell. So in ways that we are still trying to understand, and there's brilliant discoveries made every day. And that's what attracted me to viruses.
Hmm.
Uh,
You know, it's got to be puzzling to some people because we're in a department of ophthalmology and we're talking about eye diseases like glaucoma, macular degeneration, and problems with the cornea and scarring and all these other things. Where do we have someone focused on these viruses related to vision and how? Tell us a little bit about your research from that standpoint.
so I started working on, I became very interested in viruses that infect the eye, which is why I came to the Department of Ophthalmology. Uh, that really, it was an opening. There wasn't at the time a lot of research just looking at some specific viruses. And I really got interested in how some of the viruses can really cause some bad ocular damage. There's herpes simplex virus, which was a virus I'd worked on. I became very familiar with that. Actually, it was one of my family members had gotten herpes simplex virus in the eye and was having problems.
Wow.
And then at that time, before I came to Pitt, I was working on how varicella zoster virus, which causes the shingles, causes diseases, and it's a really tough virus to work with. I was one of the few people that worked on it, and I became aware of a need from the National Eye Institute to study how shingles causes eye diseases. And there was nobody in the world, well, there was one person in the world at that time who was looking at that in detail from the aspects of the virus. And I thought that was an
Ooh.
opportunity for me to come in and contribute to the eye field as well as probe how viruses cause disease.
That's fascinating. So tell us about that because and maybe I think you even have some things maybe you can show us that sort of explain that. Every time I hear it, it's always fascinating. But honestly, I'm sure as many people would have some questions on how this actually
Sure,
affects the eye.
sure, yeah. So there's one of the neat things about the virus, if I may share my screen.
Yeah.
I'd like to just show this, and we'll just go through. So I'm interested in almost any viruses that cause eye disease. And we've done some work on Varicella Zoster virus, which is my main interest. But we've also looked at how herpes viruses cause disease, how we can stop adenoviruses from causing pink eye,
Hmm.
and there's a whole host of other viruses. But this has been my big interest because it's affecting a lot of older people. So VZV is the virus that causes shingles, and you get the virus when you get, excuse me, I'm just trying to find the pointer.
Oh, it'll just be your mouse
Yeah,
is
I don't know
okay.
whether you can see it.
Yeah,
So you get chicken pox,
I can
and
see it.
the virus does something which is clever. It causes chicken pox, and then you never get chicken pox again. But the virus has traveled then down the nerve, your nerves that you feel and sense things with, and then it gets to a place called the dorsal root ganglion, and there it hides for the rest of your life.
Whoa.
And everybody that's had chickenpox, even those that have had the vaccine, the virus is hiding in your dorsal root ganglion or in all the other ganglion inside your body. And there's some people, few unfortunate people, that when they get older and their immunity doesn't last so well, the virus comes back down the nerve, it reawakens from the ganglia, comes back down the nerve, and it causes zoster, which is shingles. Now for the eye, if I could just move to the next slide. Um, so for the eye, this still happens, but there's a slight difference in that the place that the virus hides is called the trigeminal ganglion. And there that reaches to the brain and the CNS, and that comes down and causes zoster lesions. But now it happens on the face. Some of you may recognize this famous baseball coach, Tommy Lasorda. He had shingles on the face, which is a very common place. And when it's on the head, it almost all the time infects the eye and
Wow.
causes eye problems. And that's a really big problem. And
So
so
would it scar the eye, Kip? Is that what it would do?
it can scar the eye. So the first thing that happens is it gets in the cornea. But there it triggers diseases, and we don't fully understand what happens, but it can trigger nasty infiltrates into the cornea and some vascularization.
Hmm.
And this can lead to blindness. This could be very severe and lead to blindness.
How common is that, Kip? Is that fairly common?
If you're not vaccinated, you have about a 1 in 5 chance of getting zoster, and probably about a 1 in 20 to 1 in 25 chance of having it cause problems near the eye.
Whoa. Okay.
So fortunately, we have vaccines now, which is good, and it's reducing the incidence.
Hmm.
One of the things it does, though, is it affects these nerves. So one of the nerves that goes through are in your cornea. And what happens is the virus kills the nerves in your cornea, and it makes your cornea, you can't feel anything. And it's called a neurotrophic cornea. And the problem is then you may end up with a disease like this. Now, this is a disease of somebody that's had zoster, and they've had a cornea that goes totally numb. And then what happens is they poke themselves in the eye when they're doing droplets or drops or a scratch. And because there's no nerves in the eye, they can't feel it. And so it gets infected with bacteria, or you get other problems, and this leads to blindness or scarring, and that's a big problem. So that's the virus, that's the eye diseases that happen. We fortunately have a vaccine to it. The new vaccine is very good. The old live vaccine is not so good at stopping shingles, although it does work. But the new vaccine is very good, and I recommend that everybody gets it.
So everybody should get that. Do you know what age people should get the vaccine?
So the vaccine is now recommended for people over the age of 50. And that's important because most people only start to get zoster when they're over 50. And as you get older, it increases. So it rises rapidly. When you get to 65, it shoots up. And when you get to the age of 80, many people have had it twice. So
Wow.
the
Wow.
vaccine can prevent shingles. That's the one strong message I could get out there. Um, it's a good vaccine.
If
Unfortunately,
you're
a lot of
if
people,
you're unfortunate enough to get shingles and you have it and it's you know on your face just like Tommy Lasorda, is there anything you can do? Is there anything your doctor can do? Should you get you
your
know,
doctor can give you antivirals,
mhm.
um, and the antivirals are quite effective if you catch it early enough. But when the eye gets to these complications at this stage, that's when the ophthalmologist really has to scratch his head, because they've got to treat these infections. They've got to try and clear the cornea. Usually, this is an immune response to the virus. And it's difficult for the ophthalmologist to manage all the complications of zoster that can happen. There's one other thing that happens that I haven't mentioned yet, and that's pain. A lot of these patients, particularly people around when they get facial zoster, get severe pain that lasts well after the disease, the skin disease is all cleared up. And this can affect right around the eye. And it's very difficult to effectively treat the pain that you get with herpes zoster, particularly ocular pain. So those are two of the things that I want to look at. My research is, can we model how the virus goes up and down these neurons? Can we stop it? Do we know what happens when the virus is in the trigeminal ganglion? And do we know what the virus does to trigger pain? And it's been a long road, but one of my big discoveries was to model a system in which we are actually starting to model this whole backward and forward and stage of virus growth in neurons in a culture dish. So we can put eye cells on here, and this is like the cornea. We have nerves that are growing in the ganglia or on this side of a channel, and they project to the sort of corneal cells over here. We can infect the virus at the cornea, and then we can get the virus to grow up to here, go quiet for a long time, and then come back down here and model what happens during zoster. So we're starting to get a neuronal model going that is allowing us to look at the transporter virus, how the virus goes latent, reactivates, what causes it to reactivate, how it comes back down. And can we have drugs that stop it from being transported? This is all in our tissue culture model. One of the devices that we use, and you can just see it like there, it's very small. You can see it about the size of my fingernail. And what that is, that little device allows us to grow neurons and model the virus to the eye and to the ganglia and back again.
Wow.
So that's the system we have developed. And if I may share back again,
Mhm.
this is the model system in a diagram, and this is what it looks like in real life. Can everyone see that? So these are the channels, these are the nerves, on the right-hand side, these are the nerve projections coming through and the area where we put eye cells. And we're using this to see how viruses infect the nerves, and how we're also using it to see how the nerves are changed by the virus to see how it causes the pain responses, or the really severe pain responses known as post-herpetic neuralgia. And then, as I say, we're looking at how the virus can travel down the nerve, go into the nerve, and then come back down to cause shingles. And this is one of the ways that we're doing it.
That's fantastic.
So in this slide right here, you can see these little individual green dots. They're viruses. And we can actually see them traveling in the nerves. And I just wanted to show you, we actually have a movie. And this is a movie of a virus that is traveling in a nerve. And you can actually see it traveling up.
Wow.
And so we're looking at the mechanisms by which the virus travels, and also drugs and compounds that stop the virus from traveling. Because if we can use those and they're safe, we might well be able to treat someone that is at risk for shingles with something that stops the virus from coming down and infect the eye.
Wow. Wow. That's impressive.
So I do a lot of basic science research, but it really has an eye infection ending. It has relevance because I don't want people to get eye disease from Zoster.
Mhm. So has any of have you been able to work closely with clinicians and so that some of the work that you are doing in your lab has been, has helped them in treating some of their patients?
So we've got some drugs. They're we have a little bit of a problem. It's hard to test the drugs in any animal model. That's been quite difficult. But I do work with some of the clinicians. The clinicians also occasionally let me come down and speak to patients
Mhm.
that have had herpes zoster, and I really get a feeling for what trouble they're going through and how much disease and suffering they're getting. We have some new ways to stop some of the pain, and they're just entering clinical levels that we might be able to stop some of the pain that's associated with zoster and the eye infection. They're still at the experimental stage, but there is development that would stop the pain. And that's really exciting to me, because if we can get that to the clinic, we'll be, that's part of the misery of zoster, is the pain.
Wow. Well, this is interesting and exciting, and it's so different from a lot of the other work that's going on in the department, but so obviously important.
Thank
So
you.
yeah, and so you've been here a long time, and you've seen the department go through lots of changes.
Yeah, it's been
What
30
are
years.
Yeah, what looking forward in these next few years, what are the things that you're excited about for yourself and your lab, and some of the things that you hope to accomplish in the next coming years?
So we've had some exciting building times. Under Dr. Sahel, we have a lot of very young and inexperienced, but very exciting and eager faculty that are looking into some of the eye diseases. I'm hoping one of the things I can do is to help them develop their research programs. And because I've been here so long, I can mentor them and guide them, particularly as they apply for grants and funds. Um, and I'm really enjoying that. Um, in my research, now we have this model for looking at how the virus infects the nerves. we can actually look at ways to actually prevent the virus from infecting the nerves, and that's something I'm really doing now. Can we stop nerve infections? And then there's the big picture. Can we look at other viruses in the same way? We haven't done anything with virus infections of COVID yet into the eye, but there are some ocular manifestations, and it's now recognized that there are some, but not blinding, but there are some ocular manifestations that can give eye complications. So maybe we could build a program in that. I'm really excited for the future. I love, as I said, I love playing and investigating viruses, and I can apply them to eye disease and eye infections.
Well, it's I think this is an important thing for people to understand that, obviously science is the only way that we're going to be able to understand viruses so that we can understand how we can manage the symptoms that they may cause, but also understand how we can treat or maybe eradicate some of those that are causing, the most problems. So, or vaccinate people for them, and which is often the solution for them. So this is incredible work, Kip,
One,
and
one, one thing I would like to
yeah.
add is in 30 years here, I've had a great experience in collaborating with others as well. I collaborated with Dr. Hendricks just as one investigator, and we have collaborations going with other investigators here. One of the things I do is I direct a core facility, and that does molecular biology and all the techniques that I'm pretty good at or experienced in. We help other investigators to do some of their projects where they may not have the experience. That's really exciting to me as well. Something that I've enjoyed.
Is that through the Campbell Lab, Kip?
It's the whole department of ophthalmology that we've had a core grant
Oh.
that permits it's from the National Eye Institute, and this core grant supports facilities and people that run it to help other people in ophthalmology, particularly in the ophthalmology research here at Pitt, to do experiments and use techniques that they may not otherwise be familiar with or know about. Um, we've got 5 different modules, and I run one of them, which is molecular biology and bioinformatics, which is a way of looking how cells express genes and turn on genes and turn off genes. We can make vectors that allow people to express a protein in an ocular important cell under their own will. That's something I do for the molecular core, and that's very enjoyable.
Yeah, I know viruses now are used to treat, different conditions, using them in a positive way, like a Trojan horse, putting in something that you want to put into the cell and to change the cell. There's so many things we're learning, and obviously your science is contributing to that.
Thank you.
To sort of sum things up for our conversation today, tell me one thing that you would say in your long career. You're one of the senior folks on our faculty. Tell us one thing that you would say you're the most proud of in terms of accomplishments.
For my own personal research,
Yeah.
this neuro model that I did was really exciting. That was a big step forward. Collaboratively in the ophthalmology, I've loved being here for as long as I've been here, in that I've been able to work with others.
Mm-hmm.
Pitt is a great collaborative environment, and the ophthalmology department has proven to be a great collaborative environment over the time. I've been able to get experiences from other investigators. It's been great.
Yeah. Well, it's thank you for sharing your work with us today. Thank us for sharing your time, and we'll look forward to more to come for sure. And it's always a pleasure to see you.
Thank you very much. Yeah.
Okay.